Thank you very much. My name is Daniel Fung. I can speak in Cantonese, Vietnamese, Japanese, and English. Which one do you know? English or Spanish? Every year I said I was going to learn Spanish, but every year I went home and forgot about it and come back to speak English. Okay, now when I have the controls here, when I have advanced things, which one do I press? Because I want to advance. The Chinese do things backwards. So, this one. Okay, I've been around for a long time. I started doing microbiology around 1965, before your father was born. And then I observed a change of microbiology through the years. Medical microbiology started rapid methods a lot earlier than we do. I'm a food microbiologist because patients are dying in the hospital. So they are doing rapid methods much more rapid than we have. But then the food microbiology started around 1990s and started to pick up. I don't think we will ever catch up with the medical microbiologists, but there are lots of opportunities in food microbiology. So if you are interested in food microbiology, you have a good future, because I know I trained more than 100 graduate students as the major professors, and they're all over the world and doing a lot of exciting things. So I'm very proud of them. Let me see now. Okay. What do food microbiologists do? Number one, remember, you have to have good sample preparation. If not, your sample is meaningless sometimes. So you must know how to do sample preparation. One thing we love is total viable cell count. And medical microbiologists, they do not do that as much, but we must know how to do total viable cell count. Because if you don't do that, you will not know the potential of spoilage. You don't know the total count. Of course, we want to know different types of organisms. Of course, we need to know the pathogenic organisms, and we have enzymes and toxins and metabolites and biomarkers. Okay. Oh, now I don't want to go through all the names here, but how many organisms are we studying? When we started doing study salmonella as a graduate student, but now the whole there's only a list of all the organisms that we are studying, and there will be more. So your future is absolutely fantastic because there are many, many more organisms that we don't know than the ones we know. So your future will be very, very bright if you are a good microbiologist can keep on doing things. And then we are talking about examples of pathogens associated with fruits and vegetables involved in outbreaks of foodborne diseases. For a long time, nobody worried about vegetables and fruits. You worry about poultry, meat, fish, and things like that. But it's only about the past 20 years that people are really starting to realize there are lots of pathogens in vegetables. So if you are looking for the new PhD thesis, study vegetable, because there are not enough information about vegetables of all kinds around the world. Now, how many ways of doing microbiology? Just for over 5 7 weeks, 7. You should get a screen here to see what I'm talking about here. Anyway, the conventional methods is still the goal, but okay. Even we talk about all kinds of fantastic methods, you must learn the conventional method perfectly. You must know how to do pure culture isolation. If not, all your data will be meaningless. If you have a mixed culture and do all these fancy things, the result is zero. So you must know how to do pure culture studies. So that's crucial. But there are many, many ways of doing this. Oh, by the way, before I forget, the book you have in your hand is fantastic. I go to meetings all the time, all over the world. But Joseph and my mother and all colleagues made this book really, really good. So make sure you read this, keep it. It may be useful even 10 years from now, because this is all the foundations of rapid methods in this book right here that you are having. I want to congratulate you for being here to learn all these things and interact with each other. Oh, I ran a workshop a little while. I just thought it was a good idea. So I started a workshop 30 years ago, and so every year we had a workshop and we had been going for a long, long time. This just happened to be the diagram of our workshop. By the way, there's a logo here, but that is I designed a logo here with a microscope, and I just drew the salmonella. It took me 1 minute to draw the salmonella. beautiful salmonella. And now you notice the logo of this workshop has a microscope and a little grapes there. That is the Staphylococcus aureus. And I've been in this beautiful city many times, and there's a beautiful church there. That's why I have to draw a little church there. I can never pronounce that church's name, or anyway. So that is the Spanish logo. Now we have a fun time too. Well, our workshop is longer. We had the workshop for 2 weeks. And we became really good friends after 2 weeks of running around doing microbiology. But this is one of the pictures of our banquet before the banquet. That's my wife Kathy sitting there. That is our space. We always sit there. But anyway, we have a lot of people and we train several thousands of people all over the world. And we even had a medallion for people because it is such an exciting activity. But when we talk about medallion, I was really honored when I was in Finland. University of Helsinki gave me a medallion. This is a beautiful medallion, and I said, how many have you given to scientists? They said, 1 for you. So I'm going to put this on. Yeah, I'm really good at this medallion. So it's beautiful, but pretty heavy. Just for fun. Okay, let's go on. Now, why are we studying microbiology? There are actually people get sick, sometimes very sick. And I read a workshop with many people, so every time I read a workshop, I asked them, how many tests do you make per year? There was a company, they did 250,000 samples a year. Can you imagine? Just even $1 per sample, you're talking about a lot of money. Okay? And total count, this is people do a lot, but colophon count is interesting. A company did actually 840,000 colophon counts. So if you think you're working a lot sampling, you think about some other big companies that they need to run all these tests to control or to know what's going on. So we are talking about total count, colophon count, salmonella, listeria, campylobacter, Staph aureus, and E. coli. So there are all kinds of tests being done all over the world. And you may be a laboratory director of some of these things and run these tests. Okay? Well, the other thing I want to say is, do not stop learning. Even if you think you know a lot, things are moving very fast. So every time I come to this workshop, I learn a lot. As myself, I really enjoy coming here. Just these 2 lectures, we heard this morning already a lot of interesting, exciting things. We shop a lot, and my wife loves to shop, so we went to Brazil. One day she said, Daddy, I said, what? Something interesting. I said, what? There's a new coli. I said, what new coli? So I grabbed somebody from the shop, I said, well, what new coli did you find? I said, no. In Spanish, it's in Portuguese. New coli is new clothes. But believe me, ladies and gentlemen, you will find new coli, right? Learn how to detect them and kill them. Alright, now pathogen testing at the beginning, when I was doing microbiology, it took about 1 whole week to identify an organism, and half a week or 1 day. Nowadays we are talking about hours, alright? 8 hours is standard. In your time, you go down to 4 hours or 2 hours or 10 minutes, right? That's the whole idea. So we are moving faster, and there are many tests being done. Some of them do tests here. But there are some real time. There are some viable cell count we can do in 30 minutes. DAF test in 30 minutes. Other toxic tests only 10 minutes. 80%, 99% would be a target organism. But of course, you can take that out and confirm it. So we are looking at organisms doing things for us. Okay? So we did a lot of work comparing all these organisms, and with the oxalates, we can detect them much faster. This lady was doing anaerobic farming. She's very successful. She was doing anaerobic microbiology. She was not happy. I said, why? She said, so tired, so many ways of one thing. So I said, well, why don't we do it more efficiently? So we put the oxalates in. The oxalates actually help to stimulate the organisms to go very fast. And all those, we make the environment very anaerobic, so they're all anaerobic organisms also. And we also study Campylobacter, Trugina. So this enzyme is very useful. We did a lot of work to detect them faster because of the enzyme, oxidase enzyme. And actually, oxidase from the membrane of E. coli. So we can break up E. coli cells and extract this enzyme, but you can buy it nowadays. And we did non-food grade organisms, we actually did food grade organisms, so it's still very useful. We also use the enzyme for food fermentation to make organisms grow faster. And we can make buttermilk, yogurt faster, we can make sausage faster, and we did a lot of work using this enzyme to make food faster. The problem is we have to show that this enzyme is not toxic. It will cost about $1 million to do that experiment. Anyway, in the laboratory, we can make food much faster with this enzyme. Notice the instantaneous results in seconds. So we can do catalyst tests, food residue test, biosensor. You hear all these topics in this workshop. But when you are in a very famous workshop, very useful. And this book, I just browsed through it, has a lot of fantastic information here. So I congratulate you for coming here. This book will be useful for another 5-10 years. Of course, we will like to come back again a few years later to say hello. This is another story. I promised a paper in Germany to give a paper. So I was doing all kinds of things. Next week I'm going to Germany. We're going to talk about something new. And I said, okay, let me think of something interesting. Pastel pipette, of course, pastel pipette is very, very useful. So I sealed the tip of the pastel pipette, and then we put some peroxide into the other side, and put some food sampling, and shake it up, and I just use centrifugal force, the column of liquid will go to the thinner tube, and turn it upside down. If the organism produce oxygen, the catalase enzyme will produce oxygen will be trapped at the tip. So this is a 5-minute test. If you take a swab on the table, and then put it into liquid, and then put it in here, in 5 minutes you can tell whether your table is dirty or clean. If it's clean, there's no bubble. If it's dirty, you will have bubble. This test is only about 20 cents because your pipette is very cheap. So think about these things in your lab. There are some high school in Kansas right now teaching students how to determine whether the table is clean or not after you clean up the table by this little test. Very inexpensive, very powerful. And so we actually compare ribo cell count with catalase test, they correlate perfectly. So there's no question that this test is easy to use and inexpensive. A friend of mine, their company also developed something. If you have protein on the table, then something is wrong after you clean up the table. So they have this strip here, they activate it, and they just swap the chalkboard, for example. In a few seconds, if you have protein, you will see a change of color of the tip. Okay, so this is very cheap big test, but they can tell whether you have protein or not on the table after you clean up the table and whatever you want to clean up. So this is a very good test. It's called semi-quantitative test. And so of course there are many automated instruments, and you will be using some of these in this workshop, I'm sure. So what I'm telling you is, well, each one of them will take a whole lecture, so I'm just telling you that there are many instruments now that are being used that you can detect the activities of microorganisms in the environment. So you will study these few days. This is the atomic a long time ago. This is the 4-hour test from medical samples. And if you have 4-hour interaction, that means the patient's system has some higher organisms. But ATP, this is a success story because you actually can get the result in minutes, seconds, because of the ATP. The more ATP you have, the sooner you get that reaction. It's very simple, 1, 2, 3, 4, in about 5 minutes you can tell whether the surface has or does not have contamination. So that is rapid method, CRC, rapid method. And then immunology is very important. So we have all kinds of automated instruments. You will see many of them here in this workshop. Okay, and so the other speakers will talk about all these fancy terminologies and all kinds of technology, but you will touch a lot of these instruments in this workshop. And there are some instruments that can automatically do everything for you. For example, this strip here, all the reagents already in, you can just put the reagent in the machine, your sample will be sucked up by the tube, and that tube will keep going. You can clean every reaction. Totally automated in 5 hours, 4 hours, 5 hours. You say the sample contains salmonella. They are all types. You know, Listeria, salmonella, E. coli, staph toxin, Campylobacter. And so they're developing these things all over the world. And the job market is very, very good for students, for technicians, for scientists to develop. This is a success story. This is a lateral flow. If you want to detect E. coli over 5 seconds, for example, then you have some antibody that can trap the organism. And then you have some antibody that has a colored dot on the top. Then if that antibody trap your organism, and then you have lateral film. We will be doing this in this workshop. It's very simple. Well, actually, in America, they tested 2,000 samples of ground beef to give it the baseline of the occurrence of E. coli over 577. It takes only about 5 minutes to do the test. Just drop the liquid in and let it migrate. If you see one band, that is the control. If you see 2 bands, something's wrong. Problem, to find out what's going on. So these tests that you will be testing these tests this workshop. Another thing is immunomagnetic separation, fantastic technology. They put some metallic beads there, the beads that can be magnetized, and put some antibody in that incubation tube. And then you interact those antibody with the sample. If you have the target organism will be captured by the antibody on that metallic bead. Then you take a metal, a magnet, and set all the beads on the side of the tube, and then you dump the liquid away, and then you just release the magnet, and you are concentrating the organisms in the tube, bottom of the tube. And after you do that, you can do plating, ELISA, DNA, reagents, whatever. So the immunomagnetic separation really can capture your organism and concentrate your organism and make your test very sensitive. I think we are going to do these in this workshop. This is another thing. We are the first lab to do this experiment. This is a very clever system. They have a magnet at one portion of this tube. On the magnet, they put down antibody-coated beads. They captured all these beads with antibody sticking out the top of the bead. And then they put a sample and then circulate it through this system. So the first time they capture some, the second time they capture more, until about maybe 30 minutes, they will capture most of the organisms that are in that sample. So this is very good concentration. And then after you clean up the mass, then you release the magnet and you can detect the organism if you have that particular organism. So this is magnetic beads antibody, and you capture, let's say, E. coli 157H7. Then the first time you capture maybe 50%, then the second time you capture some more. So this circulating thing is actually making you capture all the potential organisms that you want to study. And after you wash everything out, release the magnet, collect the beads, and you can do the detection. This is the example of after you clean up all the things, and then you release the beads, the organisms will be detected. Very clever system. Current status of microbiology testing for genetics is very important. Genetics will actually tell you what organisms you have. So this is very important. We have DNA, RNA, hybridization tests. But those tests need about log 6 organisms to get a good reaction. A PCR, you'll be hearing this, or this workshop, you'll be running these tests this workshop. You may be able to see the demonstration in the shop somewhere. The PCR is very, very important. One thing to remember, PCR will just tell you you have DNA. They will tell you if life or dead. Who cares if they're all dead? So you still want to know the enrichment process to ensure you are monitoring living cells. Don't forget, living cells is far more important than dead cells. Okay, so what this PCR is important, but once you get them organisms that you really want to study. But there are microarray, biochip, proteomics, genomics, but important, cannot emphasize too much is you must have good sample preparation. That's the first step, right? Our sensor, of course, we can detect the organisms. but you need to concentrate them before they get to it. Genetic methods, you have DNA hybridization, PCR, you'll be working with this instrument here, your molecular beacon technology for videos and the system, viral printing, I don't know whether you may be, I think, visiting with a plant with viral printing and PubSnet systems. Another important thing is, there are two things people do. One is in this workshop, and my wife and I actually took the Concorde to go to a meeting. The Concorde 4 hours from London to New York or something like that. So I enjoy life and I work hard and I get some success here and there. I'm truly honored to be here. Thank you very much indeed.